Now we're going to move on to some discussions around microscopy. And again, some of this might be a review, but I'm hoping that some of the material that we'll cover today will break some new ground for you. And microscopy depends on light. A lot of the microscopy that we use depends on some form of light being manipulated in such a way to allow for us to view a specimen. Light bending is typically at the forefront of microscopy, where you have to move light from one medium into another. So in the case of a compound light microscope, for example, where you have several sets of lenses, we are really working with manipulating light in such a way that it gets moved in a specific direction to eventually make it up to our eye so that we're able to view a specimen. When light travels from air into a different medium, whether it's glass, whether it's oil, whether it's water, it doesn't matter, it tends to change its direction. So you'll see a lot of microscopes are very intricately designed in a very specific way to make sure that light is going to travel to where it is desired to have the best effect to be able to view whatever specimen needs to be viewed. The shape of our lenses is going to refract light at different angles. So depending on how light interacts and at what angle of the medium it is hitting, it will be refracted in a different way. And the beauty of a lens is that you can make it so that all light coming in actually gets redirected to a single focal point. And the focal point is something that you'll see often if you're looking into optometry, where if your focal point in your eye is different from how the lens of your eye or your cornea, for example, is shaped, that's when you have blurred vision. Glasses actually function in this way, where they come in and help to correct how light gets into the eye and where it'll actually focus to help to make your vision clearer. And microscopes also have a very similar mechanism, where the lenses that we use will help to concentrate light in such a way that we're able to have a common focal point and really see our specimens crisply and clearly. In the compound light microscopes that we use, we have up to 4 different lenses with different focal lengths, and the different focal lengths will allow for larger degrees of magnification. So for example, we have 4 different objective lenses on a light microscope, 4 times, 10 times, 40 times, and 100 times, and each time we increase in magnification, we're increasing that focal length, which allows us to view specimens that are smaller and smaller. We're going to talk a little bit about different types of light microscopes now, and ones with different lenses include bright field microscopes, dark field ones, phase contrast, and fluorescent. So all of these use some kind of light. Is it all white light? Absolutely not. And we're going to talk about some of those differences, of course, but all of these are examples of compound microscopes. If you've forgotten, a compound microscope simply has 2 sets of lenses, oculars that go to the eye or are also known as the eyepieces, and your objective lenses, which we can swap out for different magnifications. The way that we're able to visualize specimens under the microscope is by passing light through them. So the thinner a specimen is, the more light can pass through. The thicker a specimen is, the less light that can pass through. So we're trying to achieve a happy medium whenever we're making different samples in the laboratory, where we still have enough material there so that we can see what we need to see, but that is not so thick that you can't have any light passing through to be able to observe it with the microscope. There's a very delicate balance, and oftentimes when you're preparing, for example, tissue samples and things of that nature, you actually have a machine that will cut your tissue at very specific thickness so that when you mount it on a slide, that it is penetrable by light and that you can see through it. It's very hard, and I'm sure if you've ever cut, for example, salami or cheese or something by hand and you're trying to consistently cut those things to the same width all the time, it's virtually impossible. So we do have ways around that with different types of machines that allow us to very specifically cut a same size of tissue every time for microscopy. So this is a diagram of our light microscope. I'm very much hoping by now that this is review for you and that you're already well aware of the different components of the microscope, but this is something that is going to be very important in the laboratory, obviously, when we're talking about microscopy and viewing specimens, the most important parts here are really the objective lenses that are going to offer us different degrees of magnification depending on what we're looking at. For us to view bacteria, for example, we're always going to need that 100 times objective lens. Along with the oculars here, these are 10 times, and our oil immersion lens is 100 times. To view bacteria, you have to be looking through your microscope at 1,000 times more than what the naked eye can see. I will eventually ask you to look over the parts of the microscope and to learn them if you have not done so already, whether it's in the lecture or in the lab, of course. So just make sure that you are familiar with these. Again, I can't stress enough that familiarizing yourself with the basics in this course is really going to help when we're trying to discuss larger topics. You really do need that foundation in place to build on. This is an interesting schematic that shows the path of light through a regular microscope. So you can see down here we've got our light source here, and as the light comes up, it goes through a condenser lens or a diaphragm, which will control the amount of light that's able to get to your specimen. Eventually, it gets to the stage up here where your specimen is placed, and you can see here that's referring to this part where we do have our specimen on that stage. And then as we go up here through the objective, you can see eventually we get up to the eyepiece. That's showing that here. And then eventually up to the ocular lens where the eye is. So as we go up through the microscope, you can see that the specimen gets magnified several times, and that light is being reflected and pushed in a very specific path to make sure that it passes through the specimen and eventually makes it all the way up to your eye. This is very much a piece of precision equipment, and if any of these aspects of it get out of whack, then you're not going to be able to see what you need to see. So there's a lot of care that has to go into keeping up these pieces of equipment. So the objective lenses, as I mentioned here, are the most important parts of our microscopy endeavors, and that is because they're going to be the part of the microscope that will magnify the specimen. Yes, the ocular lenses have their own magnification, so if you pulled one off and you looked at something through your ocular lens, on its own it will magnify something by 10 times, but it is your objective lenses that do the bulk of the work. Total magnification of a specimen is a very simple calculation. It is your ocular magnification times your objective. So in this case, our oculars are always 10 times, and each of our objective lenses are 4, 10, 40, or 100. So really, you're just adding a 0 to your objective lens value, and that's how much in total you're magnifying a specimen at each respective magnification. If these lenses become scratched or broken in some way, you will not be able to see your specimen. You are also, with microscopy, looking for clarity. Your clarity is directly related to your resolution. Resolution is defined as the ability to distinguish 2 points that are close together. So when something is clear, you can usually see a lot of crispness or detail, versus when something is blurry, the ability to see 2 different points or 2 different parts of something becomes more difficult. So resolution is achieved by keeping your microscope in tip-top shape. No scratches, no dust. A big part of microscopy is making sure that we're able to actually see our cells. And because most cells on their own don't produce their own color, typically we have to add some kind of stain to them. Stains are a variety of different colors, and typically they are simple stains. Simple stains have a charge that allows them to be attracted to and retained within the cells themselves, versus the opposite. We are going to look at a couple of different staining techniques in the lab, and we're going to discuss simple and negative staining and differentiate the two. But there are also groups of cells, which we discussed last time, of course, that have their own pigmentation that do not require staining. So things like photosynthetic pigments, for example, or other types of inclusions. Inclusion bodies are things that are found inside of prokaryotes, but regardless, sometimes you can see some cellular structure without needing to stain, but not always. You can see in these cases here, these 2 examples against the blue backgrounds are photosynthetic organisms that do have that green pigmentation. These are some erythrocytes or red blood cells, which are naturally red in nature, so they also do not require a stain. And here you can see some yeast cells, which do have some intracellular structures that are visible. What those are, nobody knows, of course, but you can definitely see a little bit of cellular differentiation in these cases. And as I mentioned, we'll see more examples of these in the laboratory, but in general, we have simple stains that are going to stain the cell. They use basic dyes, which are positively charged. So the cells are negative, the dyes are positive, so the dyes get taken up and retained by the cell. Negative stains, on the other hand, are negatively charged. So what happens is the cells repel the stain and the color remains outside of the cell versus getting taken up inside. So what you end up with is a clear cell and a dark background. It's another way to differentiate the shape and structure of bacteria, and it does come with a fair set of benefits, like not having to heat fix cells or kill them in order to view the specimen. We've also got structural stains, which, as the name implies, are used to highlight different types of bacterial structures. So these are specific in some way that they will bind to and highlight different parts of the cell. Things like the capsule or flagella or the endospores. Any one of these can be exploited with staining in order to visualize them better. And last but not least, we have differential stains. The most prominent stain in microbiology is probably the Gram stain, which helps us to distinguish two different groups of cells based on their cell wall structure. A differential stain simply helps us to exclude groups of cells from one another to tell them apart based on some type of chemical property that they have. So these are some examples of some of the staining techniques that we just talked about. Here you can see our capsule stain. Capsules tend to stain clear because they are just made up of mostly sugars which don't retain any dye. We've got an example here of a simple stain where we've got the cells taking up the color of the stain itself and leaving the background nice and light. If it was a negative stain, you would see the opposite. You'd see a white cell and a purple background in this case. The endospore stain here will show the structure of an endospore. And an endospore, just to be short about it, is something that forms from bacteria when they go into a state of dormancy. So if you imagine a plant making seeds that can overwinter or can withstand harsh environmental changes, endospores are the equivalent of bacterial seeding. But instead of making and shedding something that goes on to form a new individual eventually, endospores are the cell becoming dormant on their own. So typically when you're looking at an endospore, you'll see a clear space inside. Vegetative cells that are regularly growing will be uniformly colored like these over here. But if they've started to form an endospore, the endospore is so impenetrable by stain that it doesn't hold any color. So we're going to look at examples of all of these in the lab in more detail. We're going to spend some time now talking about the Gram stain, and this was developed by Christian Gram, and it's used to distinguish two different groups of bacteria, gram-positive or gram-negative. We're going to talk more about what makes a gram-positive and negative cell positive or negative, of course, but one distinction that's important to make is that whether or not they're gram-positive or negative has nothing to do at all with what type of stain it is. We just talked about simple stains, negative stains. The Gram stain is a simple stain. It takes stains that get taken up by the cell. They're basic dyes, positively charged. That's a misconception that students usually fall into. So just be aware, gram-positive and negative have to do with the cell wall structure and not the type of stains that we're using. So this stain employs 3 different types of chemicals. We use crystal violet, iodine, and safranin. Crystal violet, when it is introduced into a cell, when it binds to iodine, it can form a complex. The complex is quite big in nature. So if you think about cells and how easily things can pass through cellular membranes, the crystal violet and iodine complex together is too big to pass through certain types of cell walls. If a cell is gram-positive, it has a really thick barrier layer that prevents that crystal violet and iodine complex from being washed away. So if a cell is gram-positive, they retain that purple color and they look purple at the end of the stain. If a cell is gram-negative, their cell wall has a lot less of that barrier component. Their peptidoglycan layer is thinner, So that crystal violet and iodine complex that forms inside the cell when crystal violet and iodine are added together can more easily escape. After it leaves, the cells are clear again, and we add a counter stain called safranin, which makes them appear pink. So I'll flip to the next slide, which might explain this a little bit more easily, but you can see in this diagram here, we add the crystal violet first, cells are purple. We add iodine, cells remain purple. If gram-positive cells are being stained, then they retain the purple color. So you can see here gram-positive cells remain purple because they have a very thick peptidoglycan layer that will not allow that large complex to pass out. But if they're gram-negative with a small peptidoglycan layer, that complex will leave. In this stage here, in stage 4, if the complex is left and the cells are clear, they can take up safranin, which is pink. But if that purple complex is still present, we do not see those cells change to a pink color. So this is going to differentiate two different types of cells with two different types of cell walls based on the same staining technique. So if you have a mixed population of bacteria, for example, and you do this stain, ones that are gram-positive will stain purple, and ones that are gram-negative will stain pink. And you might be saying, well, why is the gram status important? The way that cell walls are formed in bacteria will allow us to treat them differently. With chemotherapeutics, for example, different types of antibiotics affect specific cell types better or worse, depending on what constituents are present in their cell wall. That's one aspect. Gram-negative cells also have something called lipopolysaccharide in them, which is a fever inducer in humans. So if you have an infection and you get a fever, typically, if it's bacterial, it's going to be gram-negative because of that molecule existing on the surface of those cells. That's just a little aside. We'll talk about all this in more detail as we go. But when we talk about making slides to look at in the laboratory setting, one very important step is fixation. I did mention this briefly a while ago, but fixing cells to a slide does a couple of things for us. It allows us to kill our cells and make sure that they stick to the glass. Because if you imagine when we stain something and we add a lot of liquid to it, our stains are liquid, we're rinsing in between, we're doing a whole bunch of different things to it that involve pouring liquids on them. If those cells are not stuck in place, they're going to wash away and we're not going to be able to get a sample to look at the end of the procedure. There's two different ways that we fix our specimens. We can do it with heat, which is what we typically do in our lab, which simply involves passing our slide over a Bunsen burner flame very quickly, usually 3 to 4 times or so, and that effectively kills cells and sticks them on there quite nicely. You can also do chemical fixation, where you add a chemical to cells which causes them to kind of freeze in time and get stuck to the slide as well. There are costs and benefits to using either of these techniques. Heat, unfortunately, may change the morphology or the structure of your cells that you're observing, whereas chemicals tend to do that less. Chemical fixation tends to conserve more of the finer structures or details of cells. So depending on what you're looking at when you're trying to make a cellular prep, these are some things to consider. Other things to think about are what kinds of details are you looking for? With bacteria, typically using a negative stain or a simple stain will give you kind of the same result. You're still going to see the basic shape and arrangement of cells. The only difference is when you use a negative stain, which has a negative charge, and you get that repulsion from the cells like this, these preps don't need to be heat fixed. So thinking about what we just talked about, not needing to heat fix cells allows you to see them better in their native state. Whereas if you do a simple stain, you have to heat fix. So that's a differential that you can choose between as well when you're trying to decide what you would like to do with your samples. You view it for whatever reason, right? When you're designing an experiment or you're designing a research study, you get to pick and choose what types of procedures that you would like to use. And these are some things that you can think about. Most of the microscopy that we do is bright field. So we have a dark sample, usually fixed or heat killed, that has been stained and dyed. And most of the time we have a light background and a dark sample. So this is typically what we use with our compound light microscope. Here's an example of our eukaryotes. You can see nucleated cells that are dyed. And over here, you can see some examples of some red blood cells in the background, as well as some pop quiz time. Just kidding, of course. What kind of cells? Well, let's look at the structure. We've got rod-shaped cells, and they're in chains. So rod-shaped are bacilli, and chains are strepto. So these are streptobacilli. When we look at dark field microscopy, these are a little bit different and again have a different advantage compared to bright field. Bright field we just talked about, we have killed specimens, they're heat fixed, but dark field microscopy allows you to look at living, unstained organisms and to get a fairly good picture of them. This uses a particular piece of equipment called a dark field stop, which will force light towards the specimen only from the sides. So you'll see a different type of image. And again, depending on what you need to show when you're doing your work in a lab setting, you need to think about what type of microscopy will best suit your needs. So if you needed a living specimen, for example, you would use dark field if you had access to this kind of equipment. So you can see in this case here, our specimen's up here, and we have light coming up that gets reflected or refracted in a very specific way towards the specimen. You can see the dark field stop prevents all of this light from coming up and rather just allows concentration of light from the periphery. The images for dark field microscopy are quite striking. As you can see down here, you still see a great degree of structural detail, but you can still maintain a living specimen to observe. With phase contrast microscopy, this is another method that you can use to look at living or unstained specimens. And similar to what we see with the dark field microscopy, this has another phase plate type that can be used to achieve something very similar, where you're limiting the amount of light that can get to the specimen and controlling it in a very specific way to give you some very compelling pictures. So just to give you a bit of an example of how the different types of microscopy can be good or bad, we have the same picture here shown in 3 different types of microscopy imaging. So you see here, this is our typical bright field. These are unstained cells. Over here, this is how they would look under phase contrast. So you can see quite a lot more detail, of course. And in dark field, this gives you a totally different picture than what we get over here. So again, depending on what you want to see, what kind of specimen you want to use, whether it's living or dead, what magnification you need. These are all the things that you would think about when you're trying to choose a type of microscopy. Fluorescent microscopy is a whole other ballgame because in these cases, what we actually end up doing is making it so that our specimen emits light on its own. So we're not adding in our own light to these situations, but we're adding some kind of fluorescent entity to our sample to allow them to release light on their own. This microscopy works by getting different types of short wavelength radiation that gets released by a machine, gets absorbed by the specimen, and gets released in a way that glows. And we're able to observe that glow from the eyepiece and essentially see cells as they are tagged. This is really useful in medicine and diagnostics when, for example, we're looking to see a difference in a certain cell type in a disease state. So let's say in a normal state, we have a cell that expresses a protein, and in a disease state, that protein is mutated and is slightly different. That difference in that disease state, we can target and visualize with something like fluorescent microscopy, because you can make a tag that will bind to that structural difference in that protein if it's present. And if those cells have it, they'll bind your tag and they'll glow. So if you're looking for whether or not somebody has a disease, you can plate some cells out of theirs, and then you can add your tags in that will bind specifically to the change that you're looking for. And if they're present, you'll see them. If the person has normal cells that are not diseased, it won't stick and you won't see anything. So that's one of many ways that we use microscopy in diagnostics and also in research as well. When we're looking at different types of fluorochromes that we use for fluorescence microscopy, you will see a variety of different colors that can be observed. DAPI typically binds DNA and it'll be blue or green in nature. You'll often see FITSI or TRITSI, which are green and red. And you can see in this fluorescent microscopy here, These are some nuclei that have been stained, right? So the nuclei have lit up here with either DAPI, which is sometimes green, or you also have FITSI. And this over here looks like a cytoskeletal stain, where we have large components of the cell lighting up because they have bound a target or a tag for them that is conjugated again to that fluorochrome. Now, we don't always like to look at flat cells because what's the fun in that? Sometimes we like to see things in a more three-dimensional structure, and this is important, especially when we're looking at larger cells that have a large shape or an amorphousness to them, so that we can actually look at them in a three-dimensional plane. Because if you've ever looked, for example, at cells like amoeba that are enormous, you can never see the whole thing at the same time. If you're using your fine focus and you're trying to really find your way through different planes of depth, you're never going to be able to focus on the whole cell at once. So something like differential interface contrast microscopy uses living unstained organisms, but also allows you to take many photos of the same cells and then assemble them to create a 3D image, which is kind of neat because that way you get a true picture of the full scope of what a cell actually looks like. And it's not just a 2D flat thing that you can't quite get in focus while you're trying to do that with bright field. There's also atomic force microscopy, which is so cool. This takes a super tiny stylus that will scrape itself across the surface of things that you want to magnify and look at. So it'll scan the vertical and horizontal aspects of your specimen and will translate that into digital information to form an image. If you take a look here, these are some cells that have texture because this little tiny stylus dragged itself across the surface and it can read all the peaks and valleys and send these little bits of information to the computer. And then you end up with a very nice picture. Who would have known that cells that are super tiny are ribbed? Confocal scanning laser microscopy is another mechanism of 3D microscopy where we do take many different slices of an image of a cell and put them all together to make a final product. This is done with lasers, which is kind of neat, where you do have a lot of lasers that will get bounced around inside of the machine and allow for you to cut through frame by frame, if you will, through the specimen and eventually assemble a very nice picture at the end of the day. And here you can really see the effect of confocal microscopy, where you can definitely see depth. It's not very much because these are, again, very tiny cells, but you can definitely see the parts that are higher up, ones that are further away, even if it's just a very, very small distance, right, relative to the world that we're looking into. You're able to achieve quite a lot more detail in these types of images versus your standard bright field. With all kinds of microscopy, there are always going to be limitations. And when we consider light microscopy, its limit is at about the amount of magnification that we need to view bacteria. So you can see here, our naked eye can magnify and observe things well up to about a fraction of a millimeter. After that, we need a microscope. The light microscope, on the other hand, allows us to magnify things up to about 1,000 times what we would normally see with the naked eye. But beyond that, we really do need more help. And that's where electron microscopy comes in. These will allow us to get up to very, very small distances and be able to visualize them. Things like viruses, proteins, cellular components, individual ones like mitochondria can be visualized with electron microscopy and are super useful to us in understanding the cellular world. The difference mainly is the medium. So instead of using light, we're using an electron gun. And these electrons, very similarly though, will pass through different types of lenses to allow for very extreme magnification of very, very small samples. So the benefits of electron microscopy are a higher resolution and focusing on things that are much smaller than what we would be able to see with light microscopy alone. The disadvantage of electron microscopy is really that all your specimens are dead. There is never a time where electron microscopy can be used when specimens are living, and preparation of specimens for this type of microscopy, just like we saw with heat fixation, for example, can damage or distort the true nature of what you're trying to observe. With that said, though, we still do get a very close-up glimpse into a world that we would not be able to see otherwise. So while it's not perfect, it's certainly better than nothing. There are two types of electron microscopes. There are scanning electron and transmission electron that do two different things, and we'll look at those now. With scanning electron microscopy, we actually scan the surface of a specimen. So you're able to see some magnificent texture on different things that you want to observe. These are, for example, some pollen grains that you can see have a very nice and interesting structure to them. Who knew? And if you look at bacteria, they also have a texture. They're not just smooth little beans or smooth little balls on a microscope slide, but rather actually have some character and personality when you look at them up close with this type of microscopy. When we look at transmission electron microscopy, you're not seeing the surface, but you're rather penetrating the sample to see internal structures. So these are some mitochondria up close. You can definitely see the cristae inside and some internal structure that we would never see otherwise. And here you can see some endospores, nice and close as well. So once again, when you're choosing the type of microscopy that you might want to use, you have to consider what magnification you need, what type of structures you're trying to observe, what types of things you're trying to report on, and choose accordingly from there. And this is just showing the two types of electron microscopy side by side. Both of them are very useful in their own right, but might not always be appropriate depending on what you want to be doing. This here is actually the head of a pin. And we're going to use some extreme microscopy skills to zoom in on some stuff. So you can see here, we're pretty far away. We're zooming in a lot closer and closer still. Are you starting to see something yet? Check it out. We've got some bacteria that are hanging out on there. We're getting closer and closer and closer. And when we're right up close, you can see that we have bacteria with bacteria on them. They've got some little piggybackers that have hitched a ride to wherever these folks are going. One of the biggest problems with microscopes is, number one, they're quite large, and number two, they're quite expensive. So if you can imagine different areas of the world that might not have access to funding or the ability to use these types of equipment, there are many efforts to bring microscopy to all parts of the world, and one of these things is fold microscopy, which is a really cool initiative where you can actually get a cardboard microscope that you can actually fold up, and it allows you to be able to magnify things. It's not going to have the power of some of the microscopes that we've seen today, but it's certainly good for use in the field, good for places where there's not necessarily a lot of resources for procuring this type of equipment for a regular laboratory, for example, and they cost 50 cents to manufacture. There is a great TED Talk here if you're curious for more information, and it's just a really great way that we're bringing science to places in the world that would not be able to have it otherwise. And with that said, folks, I'm going to leave it there for now. So thank you so much for your attention today, and as always, if you do have questions, feel free to reach out to me at laura.r@nipissingU.ca. Take care.